Bismillahirrahmanirrahim, and welcome back to this next video. And this is the 18th video of the chapter 2 of Immunology. In the last two videos, I've told you about the extravasation or the diapedesis, that the extravasation or the diapedesis is actually the movement of the immune cells from the blood vessel into the damaged tissue. And the last two videos, I've told you about the first two steps of the extravasation. The first step, that was the rolling adhesion, and the second step, that was the tight binding. And I've told you that rolling adhesion, that is actually achieved by the interaction between the P-selectin and the E-selectin on the activated endothelial cells with the glycoproteins present on the neutrophils, for example, utilizing, and these glycoproteins, they are utilizing their carbohydrate part like the Lewis X to interact with these adhesion molecules like the P-selectin and E-selectin present on the activated endothelial cells. In the second part on the extravasation and the diapedesis, I've told you that there is tight binding. So when the rolling adhesion that is achieved, the endothelial cell, then they are going to express another class of the adhesion molecules which are known as the intercellular adhesion molecule 1. These are present on the activated endothelial cells, and they interact with the integrins present on the neutrophils, for example. And when the ICAM1 and the integrins, they interact with each other, the tight binding that is achieved. Now, in this particular video, I want to focus on the third step of the extravasation, which is known as the diapedesis, or which is also known as the transmigration. Now, don't get confused that here we are using the term diapedesis for the whole process. Here we are using the term diapedesis for a single step. You can find these things in the literature. In some of the literature, they describe the diapedesis as a single step. In some literature, they describe the whole process as the diapedesis. So this is not a point to get confused. So in this particular video, I want to focus on this diapedesis or the transmigration. Now, once the firm attachment or the tight binding that has been achieved, you can see over here, there are certain changes going on still in the activated endothelial cells and in the neutrophils, for example. If you look at the endothelial cells over here, the endothelial cells, after the tight binding, they are going to produce another adhesion molecule, which is known as the VCAM1. And this VCAM1 is actually the vascular cell adhesion molecule 1. So when the activated endothelial cells, they are producing this VCAM1, this is an adhesion molecule. This adhesion molecule is going to interact with another molecule present on the surface of the neutrophil, which is known as the VLA4, which is the very late antigen 4. So the VLA4 that is going to interact with the VCAM1. That means we are going towards the diapedesis or the movement of the neutrophils, for example, from the blood vessels towards the tissue. So this is one interaction. This is one thing that is happening. Another important thing that is happening at the stage of the diapedesis is that the endothelial cells as well as the neutrophils, they are producing another class of the proteins which are known as the PCAM, or which is also known as the CD31. So the neutrophils, they are producing the PCAM. The endothelial cells, they are producing the PCAM, and when they interact with each other, this process actually helps the movement of these neutrophils from this endothelial layer towards the basement membrane or into the basement membrane. So what this means is that there should be interaction between the VCAM1 and the VLA4. These two, they are present on this VCAM is present on the activated endothelial cell. This is present on the neutrophil. Similarly, there should be interaction between the PCAM present on the neutrophils and on the activated endothelial cell, so that you can move the neutrophils from the endothelial layer, from this endothelial layer into this subendothelial layer, or you can call this is the basement membrane. Now what happened is, if you can see over here, these neutrophils, they are producing these pseudopodia, and the pseudopodia, they are actually helping in the movement of these neutrophils from the blood vessel into this subendothelial area. Now, when the neutrophils, they have moved into this subendothelial layer, this is not the end of the story, because beneath these endothelial cells, there is a basement membrane. Now, this basement membrane is actually a supportive layer for the endothelial cells. Like, say, for example, if these are the endothelial cells, you can say these endothelial cells, they are residing on this basement membrane. So, this basement membrane is providing a support. So, if the neutrophils, they move from this endothelial layer, they are still stuck in the basement membrane and they cannot yet move out from the blood vessels toward the tissue. Now, this basement membrane is a specialized extracellular matrix structure, and it is made up of different kinds of the proteins like the collagen and the laminin. Now, what this means is that if you are interested in the movement of these neutrophils from the blood vessels, from the subendothelial area towards the tissue, you need to get rid of these basement membrane. So, how that is going to get achieved? Now, these activated neutrophils, for example, as you can see over here, these activated endothelial cells as well as these neutrophils, they are producing a specialized class of the enzymes which are known as the metalloproteinases. And these metalloproteinases, what these can do is that they can actually go for the breakdown of the collagen and the laminin. So, if this is the basement membrane and if you are producing the metalloproteinases, that is going to eat this basement membrane, and when this basement membrane that is eaten, that means that these neutrophils, they can move out and they can actually get entry into the tissue. So this is how the process of the diapedesis that is achieved. That the VCAM1 on the activated neutrophils, they are activating with the VLA4 on the neutrophils, the PCAM on the neutrophil as well as on the endothelial cells, they are interacting with each other, and with the help of these metalloproteinases, when you eat this basement membrane, these activated neutrophils, they are going to move into the tissue. So this is how the process of the diapedesis that is achieved. And in the next video, I'll be talking about the process of the migration, that you have released the neutrophils. Now, how you are going to direct or how you are going to migrate these neutrophils towards the tissue of the inflammation. So if you like the video, please subscribe to my channel, hit the notification bell, share it with your friends. If you have any question, you can ask me in the comment section. We will continue the discussion in the next video.